Alright, today we'll begin our discussion of biodiversity, and we're going to start by talking about three groups today. First are the prokaryotes, then the protists, and the fungi. So prokaryote is a term that you've heard before when we're talking about cells. I told you there's two types of cells. The prokaryotic cells, remember, very small and very relatively basic. It lacks any of those membrane-bound organelles. The eukaryotic cell, on the other hand, is a lot more developed, larger, and it has a lot of those organelles. So these are all slides that you saw in lecture 4, was it, when we talked about the different types of cells. And at the time, I told you that there's two types of prokaryotes called the bacteria and the archaeans. And these two groups make up the prokaryotes. But they are, in fact, distinct groups. All right, now this actually has a history to it. When you were in high school, you may have learned that biodiversity can be divided up into 5 kingdoms, right? The monarans, these are the unicellular organisms. The protists, the fungi, the plants, and the animals. And this particular scheme was developed by a man named Robert Whittaker, who was a botanist. Right? And like most macrobiologists, he wasn't particularly interested in microorganisms, and this is why he just lumped everything into a single kingdom called the Monera. But with molecular data, and using molecular data to sort of recreate evolutionary histories, some scientists started to examine the microorganisms, right? Because they were trying to create this universal tree of life. They were trying to see how every single organism was related to every other organism. And this microbiologist, Carl Woese, noticed something very significant about the microorganisms. Right? What he noticed that within the Monera there was a huge amount of diversity. So much diversity, in fact, that there's more diversity within the Monerans than in all other life combined. And what he suggested was that the Monerans should actually be divided up into 2 distinct groups that he called the bacteria and the archaea, because these 2 groups are as different from each other as all the eukaryotes combined. Right? What this means in reality is that you and a potato, a plant, are more related than any 2 species of these prokaryotes. So what he suggests is that what we need is something a level above kingdom. Right? And he came up with the idea of domain, and he suggested that all of life should be divided into 3 domains called the bacteria, the archaea, and that all of the eukaryotes should be combined into a single domain called the eukarya. Now, a lot of biologists don't really like this system because, we're comfortable with plants and animals and fungi. And so, as sort of a compromise, some biologists have proposed what's called a 6-kingdom system. So this reflects the fact that the bacteria and the archaea are, in fact, distinct groups, right? But we still preserve the 4 major kingdoms that we're familiar with. All right, so the efforts of scientists in creating this tree of life has basically allowed scientists to understand how organisms, all of them, are related to one another. And this will be the subject of our discussion for the next couple of lectures. So what are prokaryotes? Prokaryotes are pretty basic cells, right? They're the very name references the fact that they don't have a nucleus. They don't have any of those membrane-bound organelles. Right? They have a single chromosome, right? That's wrapped up in a circle. Right? And they don't reproduce via mitosis because they don't have a cytoskeleton, right? They reproduce by something called binary fission. All right, so they're a humongously diverse group. But when you look at them under a microscope, you're really only going to see 3 basic shapes. The first is the coccus, which is the spherical ones. Right? So things like streptococcus or staphylococcus, these are the spherical bacteria. The second are the bacilli. These are the rod-shaped ones. So anthrax, its scientific name is Bacillus anthracis. That is a rod-shaped bacterium. And then the last are the sporuli, which are these little corkscrew-looking things. which is kind of disappointing, right? Here's Woese saying, on the one hand, that they're so diverse that they warrant a full reclassification of all of life's biodiversity, and yet under a microscope there's only 3 basic shapes. So where is this diversity coming from? The diversity actually comes from their metabolism, right? They have a humongous chemical diversity. All right, now when we talk about metabolism, there's really 2 things that we're talking about. It's where do you get your energy and where do you get your carbon. So as far as carbon is concerned, there's 2 types of organisms called autotrophs and heterotrophs. So autotrophs are able to fix their own organic molecules by using CO2 from the atmosphere. Right? So like plants are able to convert CO2 into glucose. Right? So they are autotrophs. Organisms that can't do this are what are called heterotrophs. These are organisms that rely on already existing organic molecules. They have to eat things. The second component of metabolism has to do with energy. So where do you get your energy? Chemotrophs are organisms that get their energy from organic molecules, whereas phototrophs utilize the energy from the sun. So combining these things, you can be a photoautotroph, right? Like plants. Plants use light and then they fix their own carbon. You could be a chemoheterotroph like us, right? We rely on molecules for energy and for carbon, the organic molecules, right? And there are prokaryotes that are photoautotrophs and chemoheterotrophs, but there are also photoheterotrophic prokaryotes as well as chemoautotrophic prokaryotes, right? So their diversity comes from their chemical diversity, their metabolic diversity. Okay? So there are photoautotrophic prokaryotes that utilize light and are able to fix their own carbon like plants do. There are chemoautotrophic ones. So these are the guys that can fix their own carbon, but they use energy such as like hydrogen sulfide gas, right? If you've ever seen photos of the bottom of the ocean, the deep sea hydrothermal vents, right? There are bacteria down there that utilize the energy that's coming from that spewing molten material, right? The hydrogen sulfide gas, and then they're able to use the CO2 in order to make their own CO2 in the water to make their own biological molecules. Chemoheterotrophs is like us, right? Photoheterotrophs are light powered, so they use light as energy, but then they run around and eating things. Genetically, prokaryotes have a single chromosome that's wrapped around in a circle. Right? Now they don't have a nucleus, but there's a place where the chromosome is located, and that's called a nucleoid, kind of nucleus-ish. But they also have these little extra rings of DNA called plasmids. Right? And this is a term that we introduced when we're talking about genetic engineering. Right? These are little rings, little extra rings of DNA. They reproduce not with mitosis, but with something called binary fission. Right? The basic principle is the same, right? The bacterial chromosome will duplicate, and then the cell will stretch out and then be separated into the 2 cells. But there's no cytoskeleton in prokaryotes, and so the mechanism is a little bit different. And you can see the binary fission can be extremely rapid, and this is why you can experience extremely rapid growth in a very short period of time. So it is a form of asexual reproduction, and that by itself means that there's going to be very little genetic diversity. Right? So this is a problem when it comes to natural selection, because remember, genetic diversity is the raw material that natural selection works with. So how do bacteria actually generate genetic diversity? Well, there's 2 basic mechanisms. One is that their DNA polymerase is really bad, and as a consequence, they produce a lot of mutants. So when you have millions and millions of bacteria, there will be literally thousands and thousands of mutants. And if one of those mutations happens to be good, that will be favored by natural selection. But the second mechanism of genetic diversity is something called conjugation, or bacterial sex. It's sometimes called horizontal gene transfer, lateral gene transfer. It's all the same thing. Right? But conjugation occurs when you have a male, this hairy guy, and a female, this smooth guy. And what will happen is the male will build this conjugation tube, and then will copy its plasmid and deliver a copy of its plasmid to the recipient cell. Right? So this is not like sex like humans do it, right? Where there's two parents that will combine their genetic material to produce a third offspring that's a combination of these. Bacterial conjugation is a one-way flow. This goes from male to female only. Okay? And it occurs at the same generation. Now what's interesting about this, and only the plasmid gets transferred, not the chromosomal DNA. What's really interesting about this is that all you need in order for conjugation to occur is a male and a female. They don't even have to be the same species. So this could be E. coli, for example, and this could be Salmonella, let's say. And conjugation could still occur. Right, so here we have the donor, which is a male. Here's the recipient, the female. A conjugation tube forms, and you can see that its plasmid will copy itself, and then a copy of the plasmid will then be delivered to the female. Now, why this is important is because sometimes, remember, there are genes on these plasmids, and these genes can include things like antibiotic resistance. And what this means is that the antibiotic resistance can spread from bacterium to bacterium. Right? And remember, they don't even have to be the same species. So you can see here that E. coli by itself doesn't do anything. It's not harmful to humans. Right? Even though you hear about it in the news all the time. That's a particular type of E. coli. But normal E. coli doesn't hurt humans. Right? And Salmonella does. It makes you really sick. Right? So if this conjugation occurs and an antibiotic resistance gene transfers from the E. coli to the Salmonella, all of a sudden you have a Salmonella bacteria that's resistant to antibiotics, and that's a problem. Alright, so two types of prokaryotes here, right? Bacteria and the archaeans. And there are real differences between them. Like you don't need to know any of this stuff. This is all very biochemistry stuff. I just want you to know that there are real differences between them. Right? Differences in their cell wall, differences in their plasma membrane, just differences in how they make proteins even. We also know about the different types of bacteria, right? There's 12 different types, 12 different lineages of bacteria. Right? But I think in a class like ours, it's not important that you know the difference between a proteobacteria and a chlamydia, or a gram-positive versus a green sulfur. Like, none of that's really important in a class like ours. So we'll just say that they are a very, very diverse group. Right? And the same is true for the archaeans. There are 5 lineages of the archaeans, but again, not important for our class that we know the difference between a Cranarchaeota or Euryarchaeota. None of that's important, right? We'll just understand that there are 2 distinct groups, the bacteria and the archaeans. One thing about the archaeans that you should know, however, is that they can live in some really horrible places. Right? They're called extremophiles because they can live in places that are extremely hot or extremely cold, extremely salty, extremely acidic. In fact, scientists have found one archaean that's resistant to radioactivity. And in fact, when they tested it, it could survive 10,000 times the dose of radiation that would kill a human. Some of you may know that your farts are flammable, and the reason why is because methane comes out of your butt. Right? Now, methane is produced by a gut archaean called the methanogen that produces methane, and that's why your farts are flammable. Alright, so if we're not going to talk about the individual lineages of the bacteria or the archaeans, what are we going to talk about? Well, we're just going to discuss generally why they're important. Important for society, important for the earth, ecosystem, that kind of thing. First and foremost, they are very important agents of environmental change. Right? Now you have to understand that even though individual bacterial cells are super, super tiny, there's trillions of them. Trillions of them. In fact, if you were to weigh all the bacteria on Earth and compare that to all the other living things, right, the bacteria would vastly outweigh all other living things, all the trees in every forest, all the elephants on the savannah, all humans on Earth. Right? Bacteria are much, much more abundant than all other life combined. So, collectively, if they do something, that's going to have a very significant change. Right? And an example of this happened about 2 billion years ago when a group of bacteria called the cyanobacteria developed or evolved photosynthesis. Right? And remember, one of the byproducts of photosynthesis is oxygen. Right? So what ended up happening over time is that because there's so many of them, the levels of oxygen in the atmosphere went up and up and up and up, and it fundamentally changed the composition of our atmosphere. Bacteria are also really important for laboratory purposes. We study them a lot. In fact, a lot of the cellular processes that we know of today were actually discovered in bacteria. They also produce a lot of enzymes that we utilize. Things like restriction enzymes come from different species of bacteria. Typically, however, when we think of bacteria, we think of them as agents of disease, right? And anything that can cause a disease is called a pathogen. And truth be told, there are some pathogenic bacteria. Things like salmonella and anthrax, gonorrhea. These are disease-causing bacteria. Right? But given how abundant they are, you have to understand that most of the bacteria on Earth are not pathogenic. But the ones that are can be quite scary, right? In 2001, there was a bioterror attack where somebody was sending white powder to senators and journalists. And it turned out that this was laced with anthrax. Right? The truth of the matter is we generally don't worry about bacterial infections. And it's really strange because 100 years ago, bacterial infection was the #1 cause of death around the world. But in the mid-20th century, scientists developed a class of drugs called antibiotics. These are drugs that will kill prokaryotic cells, specifically bacterial cells. Now, the way they work is they target the differences between prokaryotic and eukaryotic cells. And it means that you can take all the antibiotics you want. It's not going to affect your cells at all. It's only going to kill the bacterial cells. And once we beat down the bacteria to a certain level, then your immune system can take care of the rest. So antibiotics as a class of drugs are effectively a miracle drug. They basically eliminated the #1 cause of death around the world. But over the last 50 years, they've become the most overprescribed drug. Right? And so that we're using it for even mild things. And as a consequence, there's been more and more antibiotic resistance. Right? The great irony is that here in the United States, we actually use more antibiotics to raise cattle than we do for people. Right? You can see that 90% of the antibiotics that we use in this country are given to cattle, right? Mostly for healthy cattle because it helps them to grow fast. The problem is that when you expose all these bacteria to antibiotics, the only ones that survive are the resistant ones. And because of conjugation, they're able to spread that resistance to other potentially pathogenic bacteria. One really horrific disease that's evolved as a result of our rampant use of antibiotics is something called MRSA, methicillin-resistant Staphylococcus aureus. So this is staph infection. Right? It's caused by this bacterium called Staphylococcus aureus. Right? And it can cause these little sores. The problem is that these can't be treated with regular antibiotics. And in fact, if you are ever diagnosed with MRSA, you're probably going to be out of commission for a couple of months. In my career, I've had maybe 7, 8 students diagnosed with MRSA, and all of them have ended up having to drop out because the treatment, the disease is so painful, and the treatment takes months. All right. That being said, like I said, almost all bacteria are fine. They're either inert or they're good for us. In fact, there are bacteria that are inside you right now. I don't want to freak you out, but you're actually definitely more bacteria than you are human, right? If you consider how many human cells are in your body, it's somewhere around 10 trillion. But how many bacteria live inside and on the surface of you? It's probably about 10 times that. So you are effectively more bacteria than human. There's bacteria in your gut that helps you break down food. When you die, you'll be decomposed by bacteria. One of the most important roles that bacteria play on an ecological level is they perform a process called nitrogen fixation. Nitrogen fixation is the process of converting atmospheric nitrogen into organic nitrogen. Right? Now you have to understand that nitrogen is hugely important for things like DNA, right? Remember, DNA is composed of nitrogenous bases, and proteins have amino acids, which have amino groups made up of nitrogen. Now, we get nitrogen from the foods that we eat, right? But we don't actually, the atmosphere that we breathe is mostly nitrogen. It's about 70 to 80% nitrogen, depending on where you are. But we can't access any of the nitrogen that we breathe. We can only eat it from the food. So how does it get from the air into the soil, into the plants? Well, that process is called nitrogen fixation, and it's done exclusively by bacteria. Almost exclusively. The other place you can get nitrogen fixation is from lightning strikes, but that's not a lot. The vast bulk of it, obviously, is from bacteria. Because bacteria are so chemically diverse, metabolically diverse, they can do metabolically they can digest a lot of things that most normal organisms can't. Right? Things like oil. The big problem with oil is when it spreads and spills all over the place, you can't really clean it up. Right? The tides that come in and out aren't going to wash the oil away because oil is nonpolar. You can use things like chemical dispersants and things like that, and that will help. But scientists have discovered a species of bacteria called Pseudomonas that can break down the oil. It's commonly called oil-eating bacteria, but what it does is it changes the chemical composition and basically emulsifies it so that the waves can break it down. So you can use these biological organisms, the bacteria, to break down chemical pollutants. In South Carolina, there was an old nuclear facility that had a bunch of, the soil was contaminated with what are called chlorinated solvents, and they're very, very toxic. But rather than digging up all that soil and transporting it to somewhere else like Nevada, what scientists did was they basically found another species of bacteria that can break down the chlorinated solvents. Right? That process of using organisms to clean up environmental problems is called bioremediation, and it's very common to use bacteria. I just read in the newspaper this morning that the scientists are looking for bacteria to break down plastics, right? Because our big environmental problem right now is excessive plastics in the environment. So if scientists are able to find a bacteria that can break down plastic, that could probably be a potential solution to our environmental issues surrounding plastic. All right. The second group we're going to talk about today are the protists. So protists are a eukaryotic cell, so they have all that fancy eukaryotic stuff, things like nucleus and Golgi and endoplasmic reticulum, cytoskeleton. But you can see that what protists are everything except the plants, animals, and the fungi. Everything that's brown up here is a protist. And what you can gather from this is that they're an extremely diverse group. So this makes it difficult because defining a protist is extremely challenging. right? And in fact, it's easier to define protists by what they are not. If you look at this list here, you can see that the protists are everything except prokaryotes. So you cross these guys out. You cross out the plants, animals, and fungi, and that's a protist. Right? So it is in fact easier to define protists by their negative. So they're not prokaryotes, they're not plants, not animals, not fungi. Now this is kind of a dumb way to describe a group because you can see that the common ancestor to all protists is right here. So the homologies that unite the protists are also going to include the plants, animals, and fungi. But because by definition we've excluded them, there are really going to be no homologies that unite all the protists and all the protists alone. So it makes them extremely difficult to categorize. Now, I don't want to give you the impression that scientists aren't aware of the relationships. As you can see from this phylogenetic tree, we do know. Right? But in a class like ours, it's not so important for us to understand the difference between parabacillid and oomycete versus a green algae versus a diatom, right? So, although scientists are aware of the different groups of protists, not so important for our class. We're just simply going to define them as not prokaryotes, not plants, not animals, not fungi. Now, this makes it extremely easy for students like you, right? Because we're defining the group so broadly, they're going to be extremely diverse. Are they autotrophs or heterotrophs? Yes. Some are autotrophs, some are heterotrophs. Are they sexual or asexual? Yes. Some are sexual, some are asexual. Are they unicellular or multicellular? Yes. Some are one, some are the other. Do they live everywhere? Like, because we've defined it so broadly, there's no real attributes that you really need to memorize. There's a very, very diverse group. There's a couple of terms that I do want you to know, though. The term plankton, if you've ever heard of that, these are the microscopic floating things, the organisms that float. There's two types of plankton, the phytoplankton, these are the photosynthetic ones, and the zooplankton, these are the ones that swim around and eat the phytoplankton. But the reason why they're important, the plankton is important, is because they form the base of the food chain in marine ecosystems. Right? There are no plants that live in the ocean, so the base of the food chain, the photosynthesizers, are protists. So, again, in terms of their diversity, there's some haploid ones, some diploid ones, some sexual ones, some asexual ones. Right? So, not really important, but extremely diverse and extremely abundant. If you look at the White Cliffs of Dover, right, it's made up of limestone. And the limestone is made up of calcium carbonate. And that rock is basically the remains of old protists. Right? The exoskeletons of these organisms are made up of calcium carbonate. Right? So like the bacteria, rather than talking about the individual lineages of the protists, we're just going to talk about them as a group and why they're important. Number one, and something I've already mentioned, and something that will absolutely appear on the exam. Their most important ecological role is that protists are the primary producers of marine ecosystems. What that means is in the ocean, they are the base of the food chain. You guys understand probably that, these organisms all eat each other, right? But they get energy and they require energy, and this is in accordance with the second law of thermodynamics. So when we're talking about a crab-eater seal, it's eating crabs and krill and all these types of things. Right? So energy is being passed from the crabs to the seal. Right? So how did it get to the crab? Well, ultimately the source of all that organic energy comes from photosynthesis. And the photosynthesizers in marine ecosystems are plankton. Right? Type of protist. You can see here how much photosynthesis is actually going on in the ocean. And in fact, it is about 50% of all of the photosynthesis on Earth. And you're going to see here that most of the high areas of primary productivity are near the shore, not necessarily in the open ocean. So this is why the fisheries are actually closer to shore than they are in the middle of the ocean. Now, the fact that protists photosynthesize, can that help us out? And the answer is yes, absolutely. You're probably aware that since the Industrial Revolution, CO2 levels in the atmosphere have just basically gone through the roof. And this is a problem because CO2 is a type of greenhouse gas, and that can trap more the Earth's radiant heat, and it can lead to global warming. So can we decrease CO2 levels in the atmosphere to mitigate the effects of global warming? And the answer is yes, if we increase the amount of photosynthesis that occurs. Right, now, this was actually a good idea that was arrived at in the late 90s. Right, and it was a result of an international treaty called the Kyoto Protocol. The Kyoto Protocol was an agreement between nations where everyone was basically going to say, we're going to cap our CO2 emissions at such and such levels. So every country chose a different level. And the idea was that if you exceeded those levels, you would pay a tax. And then that would go to help poorer nations develop cleaner industries. Okay. But the good news is, or the sort of wrinkle to this, is let's say I pledge to reduce, let's say my target is 100 million pounds of CO2, right? But let's say I took measures and I reduced it down to 50 million pounds of CO2, right? Basically, I have 50 million pounds of credit that I could sell to other countries that didn't quite make the cut. So rather than having to pay a penalty, they just pay me directly. They could buy my what are called carbon credits. So this company called Planktos got the idea of, well, if carbon is going to become commodified, maybe we could actually create a situation where we can suck up some carbon. And one of the things that they knew, these were marine biologists, and one of the things that they knew was that there was a die-off every winter of the algae, of the plankton. And it was because of iron deficiency. So what they did was they went out into the ocean, they just dumped a bunch of iron in the water, and they created this humongous algal bloom where the plankton, the algae, multiplied. And they calculated how many algae that they made, and then basically how much photosynthesis those guys would do, and then how much carbon they pulled out of the atmosphere, and they were planning to sell this on the open market on this carbon credit exchange. Unfortunately, this idea never really quite panned out because they couldn't demonstrate, like the idea is that they're going to grow more of these guys, they're going to pull more CO2 out of the atmosphere, and then because these phytoplankton only live like 3 days, when they die, that CO2 is going to end up at the bottom of the ocean. The problem is they could never demonstrate that that was the case because these guys are also the base of the food chain, so they get eaten, and once they work their way into the food chain, nobody's really sure where that carbon goes. So that was the one thing. Second thing, the Kyoto Protocol died when the United States did not adopt it. Right? The United States was the #1 emitter of CO2. So if they're not playing the game, then that's stupid. So the Kyoto Protocol lost all of its teeth. Right? So this happened when the Clinton administration stepped down and the Bush administration stepped up. The Bush administration basically said, we're not doing that. And then the third reason why this died is it turns out dumping a bunch of iron in the water is poisonous for a lot of other organisms. So they ended up getting sued a lot. So because there was no market, they couldn't demonstrate that their product worked, and their product was toxic, they basically went out of business. All right. Protists are also important because some of them are pathogenic, right? They can cause disease. If you ever go backpacking and if you have a water filter, get a good one, because if you drink out of a creek, you might get Giardia over here, and that'll give you the worst diarrhea of your life. Right? Trichomonas, right? You can just guess from the name where it lives. Right? This down here causes African sleeping sickness, Probably one protist-borne disease that you are familiar with is malaria. Right? Worldwide, it is a significant killer. We tend not to worry about it here in the United States, but around the world, it is a very, very important disease. Leishmaniasis, probably less important, but pretty gross. As you can see, it causes this sort of cauliflower ear-looking thing. And surprise, surprise, even dogs and chinchillas can get it. Here in California, you may have heard of red tides. Periodically, the water will turn red, and this is caused by an algal bloom. So this is when bodies of water get polluted with things like phosphorus and nitrogen, so basically poo and pee. All those nutrients go in the water and it fertilizes the algae, and they experience a massive bloom. And one type of species that can experience this bloom are these dinoflagellates, right, that can emit this toxin that cause a red tide. So when there's a red tide, don't go swimming in the water. Don't eat anything that comes out of the water. It's going to be very toxic. They can be agricultural pests. Some of you are aware that there was a potato famine in Ireland in the mid-19th century, right? And that was caused by a protist. And there are some good things, right? Protists are, they can live with other organisms and help them along. This is a termite, and termites eat wood, but they can't digest the wood. Inside their guts are these protists. This thing on the right, that's a protist that lives inside the gut of a termite, and that helps it to break down the wood. So that's helpful. These things are on the left, Euglenias, on the right, Paramecia. Right? You probably saw some of these Paramecia in the pond water experiment that we did on the first day of lab. Coccolithophores, diatoms. I don't know what that is on the right. Looks like an Easter egg in a hula skirt. These are diatoms. These are cool because their skeletons are made out of glass. Amoebas. Right. Now you might get the impression upon looking at those that all protists are unicellular, but this is not true. Right off the coast of California, we have big forests of seaweed, right? Kelp. Right? And these things are made up of, those things are protists. So not all protists are unicellular things. So what's the point of this group? Right? We've defined this group as not plants, not animals, and not fungi. But what do you think are the closest cousins to the plants? And the closest cousins to the animals and to the fungi? Right? They're all protists. So the important thing about the protists for our class is that they serve as these evolutionary bridges. Right? So protists are not plants, but they're cousins of plants. Protists are not fungi, but they're cousins of fungi. Right? So this right here is a slime mold. Right? You can just guess from the name slime mold that scientists thought it was a fungus. Right? But it turns out to be not a fungus, and because it's not a fungus, that's definitely not anything else, was thrown into the protist pile. Right? Green algae are the closest relatives to plants, and this is pretty apparent. Right? Up to 3 minutes ago, you probably thought that the algae in the water, the kelp in the water, was plant, but it's not. And so it's pretty apparent that the green algae are the closest relatives to the plants. Right? And same thing is true with the animals. There are protist-like animals. Sorry, animal-like protists. Right? Specifically this group called the choanoflagellates. Right? So even though protists are not plants, they're not fungi, they're not animals, there are fungus-like protists, or plant-like protists, and animal-like protists, because that's where they evolved from. Alright, and our final group today are the fungi. Right. Before we get into the sort of evolutionary features of the fungi, We're going to discuss why they are important. A couple of reasons they're important. Number one, they are decomposers. When you see a tree falling down in the forest and they have like the fungi growing out of that fallen tree, what is happening is that the fungi are actually breaking down that tree. One of the things that fungi can break down better than bacteria is keratin, right? So your fingernails and things like that are actually more likely to be broken down by fungi than bacteria. Number two, they can be pathogenic, right? So athlete's foot, things like ringworm, these are caused by fungal parasites. They are important sources of antibiotics, right? Because fungi and bacteria are often competing for the same resources. And so the fungi produce proteins that will produce compounds that will kill bacteria. And so scientists have discovered these and harnessed them and turned them into drugs. And so antibiotics are, many of them come from fungi. Right? And then they produce food, right? Both directly and indirectly. We eat mushrooms directly. This down here on the bottom left are truffles. Are a very expensive form of mushrooms, but we can also use things like yeast in order to make cheeses and breads and beer. So where did fungi come from? They came from protist ancestors, right? This is something we just discussed. They came from a group of the protists called the amoebas. Now, the fungi have had some impact on the world, right? This fungus right here is something called ergot. And ergot produces a compound that is slightly hallucinogenic, right? In fact, it's the drug LSD was modeled after ergot, so it is a psychotropic drug. This has had some important historical impacts, right? The Salem witch trials. There's a small Puritan community where the citizens started accusing each other of witchcraft. And it turns out that the year that happened, there was an outbreak of ergot in their rye, and so the food that they were eating was probably laced with psychotropic drugs, which probably explains why they were seeing witchcraft in their neighbors. Fungi are also quite important in a modern context. This is an endangered species of frog. And what is happening is as climate change occurs, the fungi become more pronounced and they're spreading their range and attacking some species, right? Including some plant species, some crop species that we rely on, but also wild species like the harlequin frog. Alright, so what are the features that unite all fungi? And this is the part where this is the important part. Alright, so there are unicellular and multicellular forms of fungi, right? So the mushrooms that you eat are clearly multicellular fungi. Unicellular fungi are called yeast. So the yeast that you use to bake bread, that give you yeast infections, these are very simply unicellular fungi. So what features do they have in common? All fungi have a cell wall made up of a polysaccharide called chitin. All fungi are what are called absorptive heterotrophs. So they rely on other organisms for the organic molecules, right? But they don't eat them like animals do. What they do is they grow into them, they secrete the digestive enzymes, and then they simply absorb all the nutrients. So that tree that's fallen down in the forest literally has fungi that's growing into them. When you have athlete's foot, the fungi are growing into the webbing of your feet. All fungi have very complex life cycles that involve a sexual and an asexual phase, but both of them involve spores. Right? So they are absorptive heterotrophs. The ones that eat dead things, the ones that parasitize other living things, both of them are growing into the matter that they're eating and then secreting the digestive enzymes. The digestion occurs outside the body and they simply absorb the organic molecules that they need. The body of the fungus is called a mycelium. and then it's made up of individual fibers called hyphae. So when you look at a mushroom, right, a lot of people think that this is the fungus, and it's part of it. Yes, it's true. But if you've ever dug them out of the ground, they're only this deep or so. This is actually something called the fruiting body. This is what allows the fungus to spread. Right? But if you were to dig underground, you would see this network of fibers. Right? They're not roots. They look like spider webs. And that's called the mycelium. Oops. And this is the body of the fungus. Each one of these fibers is called a hyphae. So this down here is the mycelium. This root-looking thing is the mycelium. That is the body of the fungus. This is simply the reproductive part. Right? And each of those individual fibers are called hyphae. And these mycelia can be massive. In fact, the largest organism on Earth is a fungus. Oops, it says CSULB, but that should say cypress. And cypress is 105 acres. So just to give you an idea of how big these fungi are. Huge. Right? Largest organism on Earth. Right, now this body shape is actually quite advantageous in terms of the fungi's lifestyle. Right? Because they're growing into the food that they're eating, and because they are absorptive heterotrophs, having a fibrous body plan is advantageous. So what's the one thing that a fibrous body has that a solid body wouldn't have? And the answer is a very high surface area to volume ratio. When your body is made up of individual fibers that are only one cell thick, right, every cell has contact with the environment, and every cell has contact with the available food source. Right? So this filamentous growth pattern maximizes the amount of surface area. It maximizes the amount of contact that the fungus has with the external environment, and therefore how much food it can secure. Fungal life cycles are actually quite complicated, right? There's asexual and sexual stages, right? And there's something in between called a heterokaryon, and none of that's important. You don't need to know any of that. The only thing that you need to know is that they both reproduce via spores. Right? If you're when you're a kid, if you've ever done a mushroom spore print, or if you've never done it, you can do it at home. Since you're it's an activity you can do at home. But there are asexual spores and sexual spores, right? But they're very, very tiny, and they're floating on the wind. They're microscopic. In fact, they're all around you. Every breath that you take, you're breathing in some fungal spores. And this is important for their lifestyle because they eat dead things, and dead things get broken down very, very quickly. They need to be able to access those resources super, super quickly. And the way to do this is to be in all places at all times, right? To have the spores everywhere so that when there is a suitable food source, they're able to colonize it. Right? So there are distinct lineages of the fungi. Right? We're not going to concern ourselves with the difference between the zygomycetes and the ascomycetes. None of that stuff is important for our class. What I do want you to know is that the differences are based on their reproductive modes. So the zygomycetes and the basidiomycetes have different sexual structures, and that's why they're different from one another. Right? So they are reproductively isolated from one another based on their sexual reproduction modes. Right? Yeah, there's lots of different types: singular, single cells, multicellular, predatory, decomposing. They do form symbiotic relationships, and I want to talk about a couple of them right now. The first are called lichens. Right? And these are fungi plus something photosynthetic. Usually it's an algal cell, so it's fungus plus the algae. Mycorrhizae, fungus plus plant roots. So lichens, fungus plus algae. We'll just call it algae. Right, now when they live together, I don't know if I have a... All right, so you've probably seen lichens before. They look like this crusty stuff on the side. What you will see if you look at them under a microscope is two different stages, or two different parts of it. Right? So remember that the fungus is made up of mycelia. And the algae themselves are, let's do the green cells, they're going to be integrated into the mycelia of the fungus. Now, algae normally live in the water. Right. But because the lichen has so much surface area, it's able to absorb enough water to keep the algal cell alive, even out of the water. And so the algal cells will photosynthesize and produce carbohydrates that the fungus part will then absorb. So because they only need each other, you'll find lichens in some really horrific places, like volcanic islands that just come out of the ground, so there's no soil even. Right? As long as the fungus has the algae and the algae has the fungus, they don't need anything else. Mycorrhizae are fungi plus plant roots. Right, so if you look at the roots of a plant, the fungus grows around that. Right? And what that does is it creates a situation where there's much more surface area to the root. So the mycorrhizae dramatically expand the amount of water and nutrients that a plant can get, because they basically help the plant increase the surface area of its roots. And in turn, the plant will photosynthesize, produce carbohydrates that the fungus part then eats. And it turns out that plants that have mycorrhizae are way healthier than plants that don't. Right? And it is in fact believed that plants could not have moved onto land without their association with fungi. Oh, there you go, right there. So they're simply way healthier with the mycorrhizae than without. Fungi also form relationships with other organisms, things like ants. Right? If you've ever been to the tropics, you'll see leafcutter ants. And what they do is they chop off little pieces of leaves and they bring it back to their colony. Now, a lot of people think that the ants are eating the leaves, and they're not. What they're doing is they're creating these big piles of leaves, and then they have these fungi that break down the leaves. They basically bring the leaves to feed the fungi, and then they eat the fungi. But some fungi and ants don't get along, right? There are, you may know that some ants are controlled by pheromones, so their behavior is cued in by chemical cues. Right? So some fungi can produce compounds that will influence the behavior of the ants. Oops, I guess this video is not going to play. Sorry about that. There's a video from BBC Earth showing something called a cordyceps fungus influencing the behavior of the bullet ant. I'm sorry, I'll send a link out for this. Alright, but I think that's my last slide.